introduce today's speaker, Guillaume Dumas. After finishing his studies in engineering, computer science, and cognitive science, Guillaume obtained his PhD in cognitive neuroscience from the Sorbonne University in Paris. And he then proceeded to do some postdoctoral research at the Florida Atlantic University in the US, after which he returned to Paris to become a permanent researcher in neuroscience and computational biology. And at the moment, Guillaume is an associate professor of computational psychiatry in the Faculty of Medicine at the University of Montreal, where he is also the PI of the Precision Psychiatry and Social Physiology Lab. And today he will give a talk entitled From Social Physiology to Social Neuro AI. Guillaume, take it away.
Thank you so much, Nadine, for the introduction, and thanks, Stephen, for the invitation. It's a great pleasure to be here and be able to mind jazz a bit with all of you and present some of old and new results. So indeed, it's a very social talk, so don't hesitate also if you have a question and something is not clear. So first, since it's social, I want to acknowledge all the people that rendered this possible, old teams and new teams, and all the fundings, of course. And by new teams, so here is like the wonderful students and postdocs in the lab with whom we try to make the bridge between this more physiological and embodied aspect of cognition to more abstract and information processing up to AI. So the lab is organized around computational psychiatry because of my appointment, but we have also this precision medicine with other discipline even beyond neuroscience and neurology and psychiatry, like oncology, for instance, using AI for tailoring personalized medicine. And the social neuro AI, we're going to be more the focus of today, which is more like the basic research part of the lab, where we try to understand the social brain in humans, and we try to carry on this knowledge to algorithms like AI. So the whole thing starts with physiology. I'm a big fan of Claude Bernard and the study of functions as a very holistic and integrative way. And old statement started this whole endeavor when in more than 10 years ago, people started to realize that neuroscience didn't necessarily covered a very important part of our human life, which is the social interaction. And especially because of technical and theoretical also limitation, there were like a sort of blank spot in the literature regarding two individuals engaged in dynamical interaction, even less more than two individuals. And that made some people metaphorically say that social interaction were sort of the dark matter of neuroscience. And for all this time, I've been like very involved in operationalizing the study of social interaction, both from a very empirically grounded perspective with electrophysiology and also through dynamical systems and computational models. So on the experimental side, mostly with a technique called hyperscanning, which is the recording of multiple brains simultaneously. And on the computational side, through whole brain simulation with biophysical models. And at the intersection between the two, through human-machine interaction, where we can either test the human through the machine being a benchmark, or testing our theory and our model by the human interacting with the model. So hyperscanning EEG was very inspired initially by work in developmental psychology. For decades, developmental psychologists have already acknowledged the importance of social interaction in the shaping of our individual cognition. And thanks to my PhD co-advisor at that time, Jacqueline Nadel, who studied a lot imitation in infants, we transfer a dual video system that was studied to that was used to study infant parents interaction to combine this with dual EEG recordings and thus have the ability to study people in spontaneous imitation during more ecological interaction. And at first, we kind of open first results at the intra-brain level, which made totally sense for a lot of people, but not necessarily in social neuroscience, which is like the neural correlate of social interaction were different from the neural correlate of social perception, which was like the vast majority of what social neuroscience was doing at the time. And especially this difference go deeper depending on the role that you're taking in interaction and the context in which this interaction unfolds. So if you are like imitating or being a model, like being leader in the interaction, you would have different network involved. And if you are like more in a spontaneous context, or like a chit chat with a colleague, or more like an induced context, which is like a forced unidirectional interaction, more like that what we have right now in a seminar, again, that involve different patterns. And typically, one of the big pattern difference is that in the forced interaction, the follower or the imitator has less social reward than in spontaneous interaction. Even if you are like the follower in a spontaneous interaction, you have kind of more reward implication. So that's why it's possible thanks to collaboration with Sophie Guillaume and Philippe Fossati in fMRI, and we replicate the symmetrical patterns in EEG and asymmetrical pattern in this context also in both fMRI and EEG. And so that has, for instance, already implication for educational science, because, for instance, peer learning would make a more rewarding experience for students than just having like classical unidirectional talk. But the most surprising and hard to swallow results that came out from these dual brain recordings were like the discovery of emergence of interbrain synchronization up to the gamma band, and I will come back to this during those social interaction. To make also something that was not clear at that time, we have like a symmetrical interbrain synchronization in the lower frequency and asymmetrical in the higher frequency. And that's already showing that it's not just because people are doing the same thing at the same time that we observe this symmetrical pattern. On top, something that was also ill understood initially is that this contrast doesn't mean the people here are doing the same thing, but more they are doing the same kind of behavior temporally. So it's like more in a jazz band when every player play a different instrument, but they are in the same groove and in the same temporality rather than doing exactly the same thing. So, well, for the low frequency in theta and alpha, it would make sense that we coordinate at the attentional level and at the sensory motor level during those tasks. But clearly the higher frequency band were hard to swallow, and I had a terrible time to publish this paper initially. We had 10 years of research on explaining this gamma band, and I will explain why finally this thing is observed. It's a question that I had during my PhD defense. 10 years later, I answer it to the examiner. So sometimes in science, it takes time to answer those kind of questions. But it's not like parapsychology. I had like the comment during my master at ENS in Paris, like, but is it not parapsychology? Or like, no. So you will see. But over the years, like other people helped to uncover other social interaction patterns. So typically with Alejandro Perez, for instance, we looked at language. Then we also looked at attachment and different kind of bonding with the team of Ruth Feldman. So where we show that, for instance, in the same participants, motor task and empathy giving task were differentially modulated depending on your level of connection with the person. So we compared friends, strangers, and romantic couples. And with some, for instance, interesting results that romantic couples were highly synchronized during the motor task, but actually were less synchronized at the neural level during empathy-giving tasks, which was not something that we expected. And now we are investigating that as a matter that romantic couple may have a internalized model of the other person. And so for more like cognitive and empathic task, then it relies less on the sensory motor coupling. So that's like already, even without the knowing the underlying mechanism, we started to look at those neural correlates in a way to bring some new theory and facts to challenge our existing theory. Of course, like also something that was first people didn't really believe in those results, but then they say, well, it's obvious because they are doing rhythmic behavior. Actually, it's not that simple either, because in other types of interaction, which are non-rhythmic, for instance, touch, and by touch, I mean without stroking, of course, we also observed interbrain synchronization, and they were like correlated to physiological phenomena, such as, for instance, the diminution of pain through this affective touch. So that was the work with Shamit Suri and my colleague Pavel Goldstein, where we looked first already to validate that affective touch can lower the pain, but also demonstrating that those interbrain synchronization may be objective measurements of those interpersonal effects. More recently, with Ruth Feldman teams, we also looked at another sensory aspect that was a bit ignored, especially during the pandemic, which is smell, also a non-rhythmic kind of sense. And here we took mothers and asked them to wear the same T-shirt during 2 days in a row, 2 nights in a row. The IRB was good with that. Then we put the T-shirt in a jar, and we had babies either interacting with their real mom or another mom wearing T-shirt of their mom or blank T-shirt. And we show that the body odor of their mom was actually replicating the level of involvement at the social level of those babies, but also raising the interbrain synchronization with the non-mom at the level of what they had with their mom. So, and still with Ruth Feldman, and even more like ironical to say that on Zoom, we also showed how those brain-to-brain synchronization may be attenuated in technology-mediated communication compared to face-to-face interaction, and thus also kind of illustrating maybe one mechanism. I'm not pretending it's the only one, but one mechanism of the Zoom fatigue that we experienced during the pandemic. And one of the hypotheses here is that those interbrain synchronization are also reflecting the self-other integration in the brain of both people. I will come back to that. And so during the Zoom meeting or through video interaction, because of the jitter and the difference of the temporality of this integration compared to face-to-face in ecological settings, our brain may face more challenge to integrate those information. And thus, like, it may account for this more draining and attentional load for social interaction on those video mediated. These like also integrate the fact that maybe like the fact to have someone in 3D really embodied in front of you may be less challenging to decode in a sense. And we are working also to control with that, for instance, and look at the time delay and the jitter in video. So like maybe there is a way to correct that actually in technology mediated interaction. So as you see, there's like a growing interest in those multi-brain recording. I started the thing in 1965 because of funny anecdotes that was associated with the comment that I got at the École Normale during my master, because like the first paper was actually a paper in Science about parapsychology, where 2 ophthalmologists, Duane and Barrett, tried to demonstrate telepathy in twins. At that time, you can publish in Science with just 1 sentence abstract and 1 photo as the data, which like show how science changed across time. Of course, this thing was never replicated and cast, I think, a big shadow on this approach of multi-brain recording. And we have to wait 2002 with people in fMRI, with especially Reed Montague, to kind of rehabilitate this approach for the neuroscience community. So I'm not a fMRI person, but so for EEG at least we get you covered. So we did like some methodological paper to help setting up hyperscanning recordings in EEG with different brands. And we have also a toolbox in Python, open source toolbox called iPipe, the hyperscanning Python pipeline that handle state-of-the-art computational method to measure those interbrain connectivity, pre-process the data, do statistics, and even visualize in 2D and 3D your results. And now, or actually also, we are supporting FNIRS on top of EEG and OPM-MEG. So we have like a collaboration with UCL actually and Robert Seymour starting on that aspect. So that's for the hardware and the software, but we need also concept to approach those things. And with philosopher, such as my colleague Meryl Verhulst, we also try to kind of decipher the different factors that play in those interbrain synchronization to try to capture what we are exactly measuring when we have this kind of average of different factors. So we brought it down to two main categories. First, the similarity of the dynamics, which can be first the behavioral level. So doing the same task, of course, will elicit similar brain pattern. This is known in cognitive neuroscience for decades. But also at the neurobiological level, having more similar or dissimilar anatomy may impact this interbrain synchronization. And so we have a lot of study going on at the psychiatry and neurological level. For instance, I'm more focused on autism, but we extended to psychosis and schizophrenia recently. And above that, we have also transcultural dimension. So we are working, for instance, with Lawrence Kiermaier at McGill on transcultural psychiatry, or where like your cultural embedding can also modify the way you are behaving and how your brain process information. So there's like a whole literature of cultural neuroscience with Shorinobu Kitayama, for instance, showing how Japanese dyads and American dyads will have different neural correlates in fMRI during social interaction, showing that the enculturation of the brain may also impact our similarity to process a similar context. And on the other side, we have communication of information. So like the more you have information exchange between two people, whether unidirectional or reciprocal, of course, their dynamic is going to be similar. I see that Chris Frith was there. They have like a wonderful paper with Carl Kristen on Duet for One, which is completely connected to this kind of reasoning. And interestingly, because I like causal loop, well, the similarity impact our ability to communicate. So typically the neurodiversity and the fact that you are departing from the neurotypical brain may impact your ability to connect and synchronize with others. But also it's through communication that we are shaping our cultural niche and establishing norms. So those different factors are measured simultaneously, and you need to have a proper design of your experiment to account for this modulation at different level. And the deal here is to try to understand what is going on. For like more than 10 years, I've been obsessed with this observation. We showed that it was statistically robust, and we wanted to show if it's just like an epiphenomenon correlate, or is it there is more to that. And so inspired by a Craver excellent book called Explaining the Brain, we look at what are the different levels of explanation that we can dig in to different methodologies. So first, it was like in echo to a TICS paper by Giacomo Novembre and Ianetti on the fact that hyperscanning alone cannot explain causality in multi-brain recording. We agree partially with that because there are different types of causality. And typically, even Reed Montague from the start in 2002 was showing that one pathway for causality is also feedback between channels. So like using neurofeedback, but at the dyadic level. So that's one alternative. Multi-brain stimulation, of course, is a royal way for doing that. But neurofeedback is also one path. But then there are also other ways to get to explanation, because I think explanation is not necessarily bound to just causality. And Daniel Kraber explained very well that. And typically from a structural causation point of view, understanding how those interbrain synchrony emerge within the brain and between the brain, and the relationship between self and other integration of information is critical, and imply other methodologies such as even classical social neuroscience paradigm with human-machine interaction. And I would give an example later on. And we can even approach those phenomena from a logico-deductive point of view with causal modeling and counterfactual demonstration using just computational models. And I will give also example with that, connecting to the social neuro AI. But for the multi-brain simulation, clearly there were like staggering results recently and coming from animal models. So in 2021, a team at Northwestern University using wireless optogenetics show that maybe those interbrain synchronization may be more than just correlates, and can actually have a downward causal action on social behavior. So they stimulate either in synchrony or asynchronously the prefrontal cortex of mice, and show that when we enforce interbrain synchronization artificially, then the social behavior of those mice are changed. And we have also preliminary results in humans, thanks to Giacomo Novembre, which show with transcranial alternative current stimulation, same kind of results in 2017, actually, in SCAN. And in our lab, we have also a setup of neuromodulation to study this causal aspect through perturbation with electrical stimulation. So that was like more the physiological part, and kind of showing how we can have a neuroscience approach of these interactional patterns in human cognition. And now the second part will be more about how we can translate that to computational models and artificial intelligence, and use those knowledge for potentially questioning also theory of consciousness, which is the topic of this seminar series. So as I say, when we observed those interbrain synchronization at first, well, we were more full of questions than answers. We were observing clearly statistically those interbrain synchronization, but we had no clue what were initially the factors. And the first computational study that we did was aligned with the neuroanatomical and biological similarity hypothesis that I show with the Merrill-Fairhurst paper. So to what extent similar anatomical brain would tend to better synchronized, okay? So for doing that, fortunately, in the 2010, I remember having a poster of Patrick Ackman's first connectome in PLOS Biology. They were like the rays of human connectomics. And I had the chance with Yasser Itoya Medina to get access to one of those connectomes matrices early on. And I set up, thanks to a computational model with copy-paste, I had 2 brains for the price of 1. I could simulate this sensory motor loop that we studied empirically. And so for doing that, I combined coupling between brain regions following the connectome. So the reconstruction through the combination of DTI and MRI of the network at anatomical level between brain region. And also I simulate coupling between brains by having like the motor regions of one brain coupled with the visual region of the other brain. So the rationale behind that is that if I'm moving my hand and you try to imitate my movements, well, we know that the motor cortex, my primary motor cortex, going to be coordinated with the velocity of the movement of my hands. And since you are watching with your screen my dynamics of hands, and you if you try to imitate that, then your visual cortex is going to be connected also with those dynamics. So in a sense, the environment is a bridge for information transfer between my motor cortex and your visual cortex, so to say. Doing that, we also wanted to connect with the empirical data. So using Maxwell equation and biophysical modeling, those simulations at the cortical level were then transferred at the scalp level to be compared with the EEG hyperscanning. And we compared the real data with simulated data. So at the resting state level, we had a pretty similar range of value, but also at the interbrain synchrony. So for a very simple biophysical model using, I forgot to mention, but here, like each brain region is simulated by a Kuramoto oscillator. And so we have we're simulating really the rhythmic oscillation dynamics in the brains. And yeah, so we have real EEG and simulated data matching on the range of value. And then we can play with something that we cannot do easily in real humans. We can change the coupling between the brain region, or we can change the coupling completely fully parameterized between the brains. So first, interesting result that was a replication from physicists studying the brain, so like Olaf Sporns and others. The real brain anatomy, so the real connectome, smooths out the transition between absolutely no synchronization and full synchronization, thus showing that the anatomical structure that was selective through evolution tends to increase the range of operation of the brain dynamics, and you're not like either dead or in epileptic seizure, but there's like a wider interval of operation. And this transition was completely collapsed to a very steep transition if you scramble this anatomy. So really like this is something known in physics. The connectome has a kind of small world topology, and this small world topology tend to smooth out those transitions. But what was not known was the interbrain aspect and how perception action coupling is facilitated first by the real connectomic structure, and second, to what extent this real anatomical connectivity tend to potentiate the coupling and the interbrain synchronization. And so that's exactly what we observe with this simulation, that first, the real connectivity tends to increase the baseline level of synchronization between the two people more than the randomized connectomics, but also that when you increase the coupling between the motor region and the visual region of the two brains, in the case of a scrambled anatomy, there is no increase of interbrain synchronization, but in the real connectivity, the real anatomy, there is an increase of those interbrain synchronization. And so that's an interesting aspect complementary to the evolutionary advantage of having a small world connectome for intrabrain information processing, which is that maybe also it has a interesting side effect, which is the ability to better synchronize with other conspecifics. Another important message here is more like from a methodological standpoint, because the field of hyperscanning is highly controversial. Like a lot of people are even questioning the scientific interest of doing that. Well, actually, if you're measuring two brains simultaneously, it's not like rocket science and interesting on its own to observe interbrain synchronization. Like here, you can see that without even exchange between the two systems, there is a baseline interbrain synchronization. And that's totally logic, because we are not measuring two quantum random number generators. So because we have two systems that are deterministically similar, their dynamics tend to be similar on average. But what is interesting here is the difference between this baseline spurious synchronization and this aspect where you have actively a coupling between the two brains. That's like the real interesting part of measuring interbrain synchronization. And so I think like that's something that is also misunderstood by a lot of people regarding hyperscaling literature, even for people doing hyperscaling sometimes. Like interbrain synchronization on its own doesn't mean anything. You need to really account for the different factors and correct for those spurious synchrony level. So these results also show that having similar anatomy may have an impact on the potentiation of coupling at the sensory motor level. And thus that helped to hypothesize that maybe in the case of neurodiversity and variation of those anatomical structure, it could impact the ability of individuals to synchronize with others, even at early stage in their life. And that's particularly connected with an hypothesis by Dimitris Bolis and Leonard Schilbar called the misattunement hypothesis for autism, which see autism not as a problem with mirror neuron or whatever gold phenotype that people are searching. I've been like working on autism for 10 years. I kind of abandoned the idea that there is like one single answer on that, but more like as a process explanation that autism is more connected to a divergence from the norm that percolate through development. And initially with having differences at the biological level, induce a divergence of trajectory at the cognitive behavioral level and also on interaction with the social-cultural embedding. And so here, our results kind of go in that direction, showing that you can have an impairment of the interpersonal synchronization without having a specific brain area defective per se, but it's more like as a general distributed phenomenon. And we're working on that with the team of Leo Schilbar using personalized connectomics. So now it's like taking one connectome per patients and per individuals and simulating either intrabrain or interbrain simulation, just like I show, and using machine learning to look at what part of the variants in the diagnostic are explainable by the anatomical resting state or interbrain synchronization. And to our good surprise, well, first, the anatomical connectivity seems to reasonably explain also and predict these diagnostic labels more than the resting state activity. But actually, the interbrain functional connectivity in those simulated dyads may actually carry more information about the phenotype at the social level and be even more predictive of the diagnostic. So we're working on that, trying to show that we can carry those digital twin ideas that come from neurology to the field of psychiatry and to autism. So another interesting aspect with computational modeling is to try to explain those weird phenomena. So Isaac Asimov was saying the best thing to hear in science, not Eureka, but that's weird. When we had this interbrain gamma synchronization, of course, we were like, that's weird because gamma synchronization is way higher than the range of dynamics of behavior. But since we demonstrate that statistically and observe that in a very robust manner, we didn't ignore the phenomenon and try to understand what was going on. So that's the thing that took me 10 years to grok. Ultimately, the answer is linked to cross-frequency coupling. So the lowest frequency, like theta, are not too much questioned because there is this well-known motor equivalence between theta band and movement velocity. So the fact that through coupling between two people at the behavioral level, you have similarity of theta dynamics makes sense. But then what about the gamma? So there is a growing literature about cross-frequency coupling where the phase of theta actually nests higher frequency like gamma. And so in that kind of coupling, and it's implicated, for instance, in working memory and other processes, the idea would be like, if we have a connection in the theta band and within brain, we have those coupling between the theta and the gamma, to what extent it can create a artificial interbrain gamma synchronization that would be like the byproduct of this internal coupling with the lower frequency interpersonal coupling. And so we test that, and that's exactly what we observe. So like that, the interbrain coupling in the theta, of course, affect the coupling in the theta. But if we look at the gamma synchronization, if there is a strong, so here the cross-frequency coupling is increasing. So here there is no cross-frequency coupling, and here we have a strong cross-frequency coupling. When we have a strong cross-frequency coupling, the level of coupling in the theta affect the level of coupling in the gamma. While if we have no cross-frequency coupling, so if theta and gamma dynamics are totally uncoupled, you can vary as much as you want the theta coupling, the level of the gamma stay the same. And also from a methodological standpoint, that's also a good take-home message for those who wants to do a hyperscaling methodology. You can see that the level of gamma synchronization actually without coupling in theta is super high. So that shows that you can have like very spurious high interbrain synchronization if you don't do your design properly. As I say, the important thing is the contrast between this and this, okay? If you're measuring that and you say, oh, I have observed gamma interbrain synchronization, I mean, those like are just methodologically spurious. So you have to be careful with those kind of stuff. But clearly you cannot have statistically significant variation of the gamma interbrain synchronization. So through these biophysical models, we kind of extend what has been done in the field of neuroscience to have computational model of neural synchronization. There is like a whole literature already on within brain, the importance of neural synchronization, starting with, of course, synchronization between spikes and individual neurons up to distance EG pair and MEG pair. So I was like in the lab during my PhD of former Francisco Varela, who worked a lot on how intra-brain long-range connectivity was more associated with access consciousness than just gamma activity. And we kind of extend that to the interpersonal level. During my postdoc, I moved to Florida Atlantic University and worked with Scott Kelso and our friend Emmanuel Tony, who passed away, unfortunately. And we tried to then carry this to a more like general frameworks. So Scott has been working on coordination dynamics for decades. It's kind of related to also some work that have been done in UCL with generalized synchronization. It's not the same language, but I think the concepts are very coordinated and aligned, pun intended. And here the point was to what extent we can use those dynamical system formalisms to carry the same analysis across different scales. So like not being bound by specific disciplines or scale of observation, and use mathematics and dynamical system formalism as a lingua franca between those different levels. And in the context of coordination dynamics, the idea is that clearly we have either a way of describing things in matter of components of the systems, or we can use it also to describe patterns, so synchronization or anti-phase, in phase, and those kind of stuff. And we can do that across different scales. So carrying this in mind, and now I'm affiliated to MILA, the Quebec AI Institute, where deep learning was developed partially with Yosha Bengio. I try to make a point with my colleague in computer science that AI, on top of having to take inspiration for biological plausibility, which was the point of deep learning compared to good old-fashioned AI, should also account for temporal dynamics, because for now it's very feed-forward and discrete in a way of processing information, but also account for social embodiment and the fact that we have maybe a solipsistic approach of artificial agents, while we are social beings that are interacting with others, and that shape our cognition as well. So that's what we try to describe as this social neuro-AI roadmap. And one of the motivation partially was also aligned with, for instance, the work of Michael Graziano in his book, Consciousness and the Social Brain, that, well, the same brain region that are computationally processing in the social context to attribute awareness to others may also be the one that we are using for attributing awareness to ourselves. And like the link between metacognition and more like theory of mind and approach to other being. So getting interested to that, there were like someone saying like nothing in biology makes sense except in the light of evolution. In my previous appointment to Institut Pasteur, I dive into paleogenomics. I'm very enthusiastic with very new stuff. So like, for instance, at that, I mentioned the connectomics in the 2010. In 2020, there were like the burst of archaic genomics with Svante Pavo, who got the Nobel Prize last year. And we had for the first time the DNA sequence of Neanderthal, Denisovan, etc. And I used this advantage to be in a genetic lab to carry on a systematic analysis of human brain evolution in the recent primate lineage. And so to try to understand what in the recent Homo sapiens emergence was connected to social behavior and social cognition, and the relationship between what we just described regarding awareness and complex high-level cognition. And so we looked at first the whole genome systematically, and to our big surprise, like that's completely like the opposite of what we expected, the brain appeared as one of the most conserved tissue in the whole body. So you don't mess up with the brain. Actually having mutation in brain-related genes tends to have birth defect or a very cognitive impact. So it's not like a tissue where you can have easily mutation. At one exception, the olfactory receptor, which are like known for geneticists for years to be very fluctuating to adapt to the environment and new smell or new food. So there's like a relaxation of evolution for the olfactory receptor, but the rest of the brain is pretty conserved. But we can still look at what are the most divergent genes in the Homo sapiens and what are the most conserved, and look at where those genes tend to accumulate in gene expression in the brain. And so thanks to the wonderful public data, such as the Allen Brain Atlas that show where brain genes are expressed, and the NeuroVault, which is like a gigantic statistical database of a bunch of fMRI studies with the beta maps. I did a kind of comparison of where those evolution tends to aggregate at the genetic level in the brain, and to what extent those brain regions correlate with task in fMRI. And we show that there were like more than chance enrichment of social task and typically language, and something that we put initially in the paper but didn't make it to the final cut. We show that there is like an accumulation in the Broca area of those divergent genes actually. So that's like a perks of message in the bottle if some people are interested with that. And well, from the evolution of humans, it makes sense to have those advantages at the social level, because the characterization that was known for since the 70s is that a characteristic of the Homo sapiens brain compared to other primates is that we have a delayed critical periods of plasticity, something called neoteny, that increase our window for social learning. So we can grasp more plasticity from cultural environment and education, and thus having like a slight divergence on those genes implicated in social cognition may have helped in this enculturation phenomena. But so for now, it's more like a correlation statistically on 3 archaic hominids and some primates. So to test that, we went to the more like counterfactuals and logical deductive. And with Jean-Pierre Changeux, we published recently this computational model of the global neural workspace But this time, compared to other models that have been published before, for instance, with Stan Dehaene, we integrated first plasticity. So instead of having the hardwired global neural workspace, in this paper, we show what are the necessary biological mechanisms to establish through development a working global neural workspace. And second, we try to have these nested levels of information processing, with the last one being the metacognitive level, aligned with some work at UCL actually with Emmanuel Shea and Chris Frist, saying like that's the metacognition aspect that is missing in the global neural workspace. So we are clearly going in that direction at the biophysical level. And for a more abstract level, we have also another project with Yoshua Benjo and more the deep learning people to try to see at the computational cognitive level architecture, but not really with the details of the biophysics implementation, how we can grow an attention schema in the brain, whether like we call it attention schema or more like a controller of attention at a higher level, depending on which people you are talking about, the wording are different. But here, the idea is to have this metacognitive loop that we have here from a pure functionalist approach embedded in a deep learning model, and show in the same way that it was shown by Wilterson and Graziano already, when you backed up directly the architecture in an hardwired way, you give a computational advantage. To what extent we can learn those attention schema and those feedback loop. And I see there is a Axel Clearance also here. We have a similar approach inspired by Axel's work on feedbacking, making a feedback loop at the higher level, and connect also with your work, Steve. Like I tend to be a lumper and not being bound to one theory, so I take the tools in the theory that I like. But so the advantage with deep learning modeling here is that compared to biophysical modeling, which is very heavy and computationally intensive, we can run a lot of environment and a lot of tasks, including multi-agent reinforcement learning tasks. And we can test this hypothesis brought up by Graziano in the consciousness in the social brain, to what extent cooperative task and social task may have carry-on effect on individual ability at the high level metacognitive aspect. So like we push those agents in a social framework, and we look at then their individual property at the cognitive level and see to what extent we can accelerate artificially the evolution in silico and reproduce this pressure from evolution at the social cooperative level. So then something I mentioned with the link with the work with Scott, we have also the temporal dynamics part, which ultimately is not yet embedded in this range of models, but we have a big survey already. If you're interested, we took a lot of time and pleasure to do those figures to kind of cast and draw the landscape of what is at the intersection between machine learning, neuroscience, and dynamical systems, and to what extent we can put more dynamics in AI. And we found a lot of interesting approach. In our case, we explored one called scientific machine learning, where you insert computational models in deep learning architecture. And we show that, for instance, combining both deep learning and computational model of human brain activity, we can outsmart most of the state-of-the-art methods in forecasting brain activity. So that's a interesting toolkit, not even just for social neuroscientists, but in general. But it's also my colleagues in computer science are very pragmatic, so they want to show, they want proof that saying like when dynamical system can be useful for AI, they want benchmark and proof of that. So that's one example. And from a more cognitive science perspective, we also use this dynamical system modeling with Scott Kelso in a paradigm we call the human dynamic clamp, in homage to the dynamic clamp in electrophysiology, where a human is coupled in real time with the dynamical systems of a human. And here, that's what I alluded in the beginning. We can use that for both testing our model. If the model, for instance, passed the nonverbal Turing test, we are considering that the model is pretty good. But we can use those kind of paradigm to test the human. In that case, we can do neuropsychology and psychophysics of social interaction in clinical settings. So funny first behavioral results regarding the nonverbal Turing test. So having a computational model interacting with the human, we can fully control to what extent this model is cooperative or competitive and the difficulty of the task. And funny results that we found using this Turing test. First, indeed, the humans are fooled, but on top, they tend to blame a lot the other virtual partner, the partner, especially when the task is more challenging, even if the cooperation of the partner is the same. So like, it's very funny because instead of assuming that the task is more complicated, they tend to blame the other person to be less cooperative. So it's a very funny social psychology results that we ended up with that. And combined with EEG and brain recordings, we had also the ability to look at those self and other network that we mentioned earlier and show that the RTPJ area that popped up in a lot of social neuroscience results, but also in those interbrain synchronization as a hub and where the strongest are those interbrain synchrony is actually the region where the network associated with self-behavior and other behavior are tightly integrated. And so that makes sense to explain, that's the more logical, not logical deductive, the second type of explanation to show that you have interbrain synchronization here, because if you have self and other information there integrated, it's symmetrical between the two people because the self had to become the other and the other become the self. And so that makes sense that this brain region end up being the most robust hub for interbrain synchronization. We also show that this hub on the posterior level tends to be more interacting with the frontal regions during the evaluation of cooperation or attribution of intention to the other agent. And we try indeed to make the bridge between this sensory motor imitating embodied literature and more like representational metacognitive and more like higher level abstract social cognition in the term of also Kahneman and computer scientists, like to bridge the system 1 and system 2 of social cognition. So briefly, because I see we are heading to the end. So those system 1, system 2 types of interaction can be probed with this human dynamic lamp. So we can look at how the human coordinates at the motor level, but we can look at also how he inferred properly the intention. And so we use that in clinical settings as a way to, at the behavioral level, also predict diagnosis of autism. And we have even better prediction just with behavior and 15 minutes interaction with this avatar. And well, the problem is like kids in general and kids with autism are pretty direct. So they immediately say that my video game sucks. So I moved to video games design to gamify the experiment. And now we have even a version of this paradigm running in mixed reality. So that's look like that. So you have different balloons and avatars and you interact with them. So fortunately, Montreal is a big capital for video game industry. So I had a lot of help for the game design and so on. And ultimately, you have the participants playing this video game in mixed reality while the clinician or the parent can monitor what's going on. We can have even a feedback on if the kid is stressed to interrupt the experiment if it's the case. And while the kid is gently playing this video game, we are collecting a bunch of data from the movement, the reaction time, the eye tracking, the heartbeat, and so on. And we can use those different information through classical dimensionality reduction or more advanced deep learning methods to plot profile of those individuals. And since video games are able to be scaled up, we can collect large normative shards of those profiles to show at the individual level where you are compared to the controls. And the idea is when you have normalized those profiles, you can do stratification and go to a homogeneous subgroup of patients and redefining potentially the taxonomy of the labels that are in the DSM. Right now, we are validating that, but ultimately, there is a lot of similarity between the being in the zone and the flow in game design and the proximal development zone of Lev Vygotsky. So we are trying to design video games that tend to steer the proximal development zone of those kids through reenactment of the development that may have been altered. And as a final thing, so I mostly talked about the integrating neuroscience here with the moving from one trait to a whole phenotype, integrating social interaction and neuroscience. But we have also in the lab, as I say, like the more precision medicine and computational biology side, working with geneticists, oncologists, and so on, and which is like the traditional pathway for precision psychiatry in the literature. So like a lot of the people doing precision psychiatry are more doing genetics and trying to tailor psychiatric help with the genetic profile of the people. And in our case, we try to combine this pathway and this pathway. Fortunately, there are like changes in the community in mental health, and typically questioning the DSM taxonomy to move to more dimensional, for instance, description, such as the research domain criteria. But the problem is that the research domain criteria still lack the social dimension in those dimensions. So it stopped at the individual. And I learned that it's partially caused by the funding structure in the US, because the social dimension depends on another agency that the NIMH. So that shows sometimes how funding structure may impair our ability to do science. But if you are interested with this and how to bring those interpersonal and interbrain methods to psychiatry, I have like this short piece recently describing that. So at that time, I put interpersonal psychiatry, but with Dimitris Bolis and Leo Shiva, we went even saying like interpersonalized psychiatry. So bridging the gap between interpersonal and personalized psychiatry. And to show also something that I think for UCL, it's important to mention that, because you have been like really at the forefront of computational psychiatry in a sense with a different approach. To show that computational psychiatry can go beyond individuals, not only at the dyadic level, but we can use computational methods to do policy making for psychiatry and so on. This is my pandemic paper when we didn't have the opportunity to do empirical studies. So we use Twitter and PubMed to look at discourse, like popular discourse and scientific discourse around autism. And we show that there were like a total misalignment with the topics that interest the stakeholder and what is funded by and carry on by the biomedical research. So showing like that can be also important for computational psychiatry. And another example, and I finish on that because you are in UK, is to show also that we can look at social determinants of mental health using computational methods. So this is like a collaboration with Cambridge and Stanford, where we took like the whole population of UK. So it's like the first time ever that I did descriptive statistics and not inferential statistics. The linear model ran for 3 weeks on the supercomputer. We carry out a statistical analysis on what are the social determinants at the ethnic and geographic and socioeconomic status level that modulate the access to diagnosis of autism, and showing that among the investments of, for instance, policymaker pushing a lot of money into the genetics and the more like traditional biomedical research for the stakeholder, actually investing in having a hospital near the people tends to have a gigantic effect size in their ability to get a diagnosis, and showing that social science and those social determinants are super important and maybe neglected because they have been maybe not as computationally and quantitatively assessed than genetics and other biological things. And so the policymaker tends to follow more like quantitative stuff. But that's also an opportunity for computational psychiatry to get this type of information out to policymaker and have real impact for stakeholder. So that's like a wrap up. Sorry, I mean, we had like just in time. There's not so much time for question. I can stay a bit more, so but that's about it. Thank you very much for your attention.
All right, that's all on mute, and thank Guillaume for a great talk. Thank you so much. Super interesting. Yeah, so we do have an hour and a half, so we have quite some time for